Thank you for taking some time today to learn more about the nCounter analysis system for the analysis of formalin-fixed and paraffin-embedded samples. I'd like to spend the next few minutes describing how the nCounter technology can assist you in analyzing the difficult but clinically important sample types such as FFPE. In today's webinar, we will first review the promise and challenges of utilizing FFPE samples in your gene expression research. Next, we will highlight the advantages of the nCounter system in analyzing nucleic acids from FFPE tissue. And finally, I will provide some examples how the performance of the system on FFPE samples and compare it to other methodologies such as qPCR. FFPE samples represent a priceless resource of characterized samples for the clinical research community. Their value lies in the biochemical and clinical information that is preserved in this unique collection of samples. For example, millions of FFPE tissues are stored in biobanks worldwide. The tissue histology and nucleic acid molecules are largely preserved. A large variety of cancer tumors and diseased tissues have been collected over the years, and often these collections have detailed data associated with the sample, and patient outcomes are often known. Many collections have longitudinal data and long-term storage that allow for retrospective testing. This enables testing of hypotheses over months rather than years. Based on these characteristics, FFPE sample collections offer enormous potential to further scientific knowledge and translational research. Despite the number and value of these FFPE resources, there are multiple factors that have made genomic and molecular analysis of the FFPE samples notoriously difficult. For example, the fixation times and methods of collection are not standardized and can often vary significantly between or even within institutions. The age of the collected material and specific storage conditions may also vary widely. RNA and DNA isolated from these tissues are often degraded and contain modifications that can inhibit downstream enzymatic steps. In addition, the amount and quality of cellular material contained within a single FFPE slice often leads to low yields of extracted nucleic acid for many samples. Taken together, the variability introduced by this combination of factors often introduces noise into the molecular measurements and confounds the interpretation of the results. The nCounter technology offers a simple and effective way around many of the issues associated with analyzing FFPE material. First, our technology measures RNA or DNA target molecules directly with no target amplification or PCR. Thus, there is less impact of chemically modified bases on the results. Second, the target region required for detection with the nCounter system is approximately 100 bases. This, as you will see, allows for a good deal of tolerance in the amount of RNA or DNA degradation for a given FFPE sample. Third, the assay is capable of measuring up to 800 genes in a single reaction, minimizing sample inputs and vastly decreasing the number of sample handling steps required to obtain data on many genes. And finally, the workflow is simple and hands-off. After a straightforward hybridization setup, subsequent sample handling is automated, reducing possible workflow errors and making it attractive to clinical research environments. For these reasons, the nCounter system has been widely adopted by the cancer research community and is currently being utilized for a variety of high-impact research studies. As mentioned previously, the nCounter chemistry was designed to measure hundreds of nucleic acids directly without the need for amplification in a solution-based hybridization. Here's how it works. For every target of interest, we design 2 adjacent 50 base pair sequences that are complementary to a 100 base pair target region. One of the target-specific oligos is covalently linked to a biotin moiety to create what we call the capture probe. The other target-specific oligo is covalently linked to a color-coded molecular barcode to create what we call the reporter probe. A solution-based hybridization is performed in the presence of excess probe to drive the reaction to completion. Up to 800 targets can be hybridized in a single reaction tube. The hybridized complexes are then purified via an automated liquid handling robot called the prep station and bound to a streptavidin surface for imaging. The purified hybridized molecules are then elongated and immobilized in order to read the barcode by the digital analyzer. The scanner then digitally counts the number of barcodes hybridized to a single RNA or DNA molecule for each target. Again, note there is no amplification, and therefore a single molecule equals 1 counted barcode. The nature of the digital data allows us to maintain high precision and sensitivity over a large dynamic range. The image analysis algorithm processes the 4-color images and counts individual barcodes. On the left is an overlaid image of a single barcode, and on the right is a section of a single field of view containing many barcodes for many genes. It is important to note that the barcodes are counted, and it is not based on the fluorescent intensity of each spot, like analog technologies such as microRNAs. In addition, specific criteria for counting barcodes results in a very low false positive detection rate of around 0.1%. All purification, sample loading, and scanning steps are automated and handled by the prep station and the digital analyzer. In the next few slides, I'll show you some nCounter data from FFPE samples. As mentioned in previous slides, nucleic acids from formalin-fixed and paraffin-embedded samples are often degraded. This slide shows the size distribution of an RNA isolated from FFPE tissue on an Agilent bioanalyzer. The maroon line shows a typical total RNA size profile where 18 and 28S ribosomal RNA bands are clearly visible and intact. The green line is RNA extracted from a frozen slice, showing a similar profile, indicating the process of slicing and isolating the RNA from the frozen block does not itself degrade the RNA. The blue line is a bioanalyzer trace of RNA extracted from an FFPE sample from the same tissue sample. The 18 and 28S peaks are now clearly gone, and the RNA is distributed over a wide range of smaller sizes. Note that this is a single FFPE sample, and RNA size distributions can vary widely across samples based on tissue types, fixation methods, and time of storage, etc. In order to quantitate the impact of degraded RNA samples in the nCounter system, an experiment was performed on a set of RNAs that were chemically degraded for various times. Human reference and human brain samples were degraded and run on a bioanalyzer to determine the degree of degradation. This graph clearly shows the decrease in the RNA size profile with increasing times of degradation. The degradation profiles cover the range of most FFPE samples. An RNA ladder and a control total RNA are also shown. At 5 minutes of degradation, both the human ref and the human brain samples are largely below 500 bases in average length. We then examined what the impact of this amount of RNA degradation would have on the nCounter system performance. RNA from each time point was hybridized with the nCounter code set and compared with intact total RNA. This graph shows the overall correlation in the gene expression profile based on counts. You have excellent correlation with an R-squared of 0.99 even with RNA that is degraded for 5 minutes. The slope of the line indicates the average loss of signal across the code set due to this degradation. At 5 minutes, 35% of the signal was lost in this sample. Despite this loss in signal, the correlations are still maintained. These results indicate the nCounter technology is relatively insensitive to RNA degradation and ideally suited for FFPE type analysis. Most gene expression studies, however, are designed to look at the differences between two biological samples. Therefore, we examine the correlation and fold change between the human ref and human brain samples for both intact and degraded RNA. This graph shows a log2 plot of fold change values for the human ref and human brain control shown on the x-axis relative to the various pairs of degraded samples shown on the y. Again, we observe excellent correlations across the code set between the degraded and the intact samples, even at the 5-minute degradation point, which is approximately 500 bases. The results further support the utility of the nCounter system for gene expression analysis on degraded samples such as FFPE. This slide shows an example of the correlations in expression values or counts for two actual FFPE samples, in this case liver and lung, relative to intact matched total RNA control. As observed with the chemically degraded RNA samples, the correlations in overall expression profiles remains high with an R-squared value of 0.91 and 0.96 between FFPE and the matched control RNAs. Again, these correlations are maintained even though there is a loss in signal due to the target molecules being degraded. We have also been investigating methods to make the analysis of RNA from FFPE tissues even easier for our customers. A protocol was developed to measure RNA directly from crude FFPE extracts without the need to purify RNA over columns, etc. Briefly, the method consists of a deparaffination of the FFPE slice followed by protease K digestion to remove the proteins. An aliquot of the protease K extract was then used as the input to an nCounter hybridization. As a control, the rest of the FFPE extract was processed to extract the FFPE RNA. In this example, we show a scatter plot of the expression values for a heart FFPE sample relative to a matched total RNA control. The green dots show the counts for an FFPE RNA from this sample relative to the intact RNA control. As we have seen previously, the correlations are high despite a moderate loss in signal due to the degradation. The orange dots show the results from the FFPE extract RNA from the exact same sample relative to the intact total RNA. These two data sets are largely overlapping and nearly identical. In fact, if one compares the FFPE extract directly with the FFPE RNA, shown in the inset at the bottom right-hand side of this figure, the correlations are nearly perfect, indicating the extract provides the same information as the purified RNA. Thus, if the study design allows, using FFPE extracts may save considerable time and money while providing great performance from FFPE tissues. Besides RNA degradation, RNA isolated from FFPE samples is also chemically modified due to the fixation process. While many of the commercial RNA extraction kits for FFPE contain steps to reverse these modifications, they are not 100% efficient. Many labs have shown that the modified RNA from FFPE samples can inhibit enzymatic manipulations, including reverse transcription and qPCR. In this experiment, we performed gene expression analysis of 5 genes using either the nCounter system or quantitative PCR. We then measured the ratio of absolute signal, either in counts or CT values, between the FFPE RNA and the matched total RNA sample. As shown in these two graphs, RNA isolated from FFPE tissue has a larger negative impact on qPCR than it does in the nCounter assay. In all 5 genes, in 2 separate samples, the loss in signal between FFPE and matched RNA is greater in PCR, shown in the orange line, than for the nCounter system in the green line. Also note that the loss in signal for individual genes via PCR can be 10 to 50 times greater than that of the nCounter system. Presumably, this difference is due to inhibition of the reverse transcription step due to the presence of the modified bases in the template RNA. These results suggest that when examining many genes over many FFPE samples, the nCounter system will outperform qPCR when reproducing results from frozen tissues. The difference in performance between nCounter and qPCR and FFPE samples can have a profound impact on your experimental data. A recent peer-reviewed paper from the University Health Network in Toronto has directly compared gene expression profiles of 19 pairs of matched flash-frozen and FFPE tumors by both nCounter and qPCR. The figure on the left shows the nCounter data, which had a Pearson's correlation between all FFPE and fresh frozen samples of 0.90. The figure on the right shows the same experiment for qPCR, which had a correlation of just 0.5. The authors conclude that the nCounter system is a superior solution for gene expression analysis of their FFPE samples, especially when it comes to large-scale validation studies. In the last few slides, I will review the performance of the nCounter microRNA and CNV assays on FFPE-derived RNA. In recent years, microRNAs have gained an increasing role in cancer in both the biology of the disease as well as potential biomarkers for cancer. NanoString's microRNA panels are also well suited for measuring the abundance of microRNAs in FFPE tissue samples. This graph shows the correlation in log2 counts for microRNAs between the total RNA from a frozen tissue and total RNA isolated from a matched FFPE sample. As we have seen previously with the gene expression assays, the correlation between frozen and FFPE results is excellent, with an R-squared of greater than 0.96. Thus, similar to the gene expression products, the nCounter system microRNA panels perform equally well in FFPE samples. For many research questions, analysis of RNA alone does not provide adequate information. For example, alterations in the number of copies of a gene or regulatory element in the genome can also be important drivers of a disease or phenotype. We have recently developed a protocol for analyzing DNA from FFPE tissue on the nCounter system. In this slide, we have examined the expression of 85 genes that are known to exhibit changes in copy number in some cancers. The graph shows the copy number measurements for DNA extracted from a matched pair of frozen and FFPE tumors. In tumor 1, shown in the upper graph, examples of homozygous deletions, or 0 copies, and duplications at 4 copies were detected in both sample types. In tumor 2, in the bottom graph, 2 genes were detected with greater than 20 copies relative to a diploid cell line. Again, the matched FFPE and frozen tissue are in good agreement, demonstrating the dynamic range of copy number capabilities of the nCounter system. In summary, I'd like to close by emphasizing that the nCounter analysis system is ideally suited for the analysis of nucleic acids from FFPE tissue. The nCounter system enables a high-precision analysis of 800 targets from FFPE tissues in a single reaction. The FFPE performance for all the nCounter assays, gene expression, microRNA, and copy number, is excellent. The system is easy to use and is automated, and many of the results I have shown you today have been confirmed by independent analysis. For these reasons, a large and growing base of leading cancer researchers trust nCounter as the gold standard for analysis of archived tissue. Thanks for your interest in NanoString technologies today. Please contact us if you have additional questions.